Now let's start by learning about lipids in detail. First, we will be learning about the classification of lipids. Lipids are a diverse group of organic compounds that are insoluble in water but soluble in non-polar solvents such as chloroform, ether, and benzene. They are the important class of biomolecules found in living organisms, and they have a wide range of functions such as energy storage, membrane structure, and signaling. There are several ways to classify lipids, but one common way is based on their chemical structure and properties. Here is a detailed classification of lipids. First, we will be classifying it as fatty acids. Fatty acids are the simplest type of lipids and are composed of a long hydrocarbon chain with a hydroxyl group like COOH at one end. They can be saturated or unsaturated depending on the presence or absence of double bonds in their carbon chain. Saturated fatty acids have no double bonds, while unsaturated fatty acids will definitely have one or more double bonds. Fatty acids are the building blocks of many other types of lipids. Next is the triacyl glycerol. Triacyl glycerols, also known as triglycerides, are the most common type of lipids in living organisms and are the main form of energy storage. Triacyl glycerols or triglycerides. They are composed of three fatty acids esterified into a glycerol molecule. Triglycerols can be saturated or unsaturated depending on the fatty acids that make them up. Phospholipids. Phospholipids are a type of lipid that are important components of cell membranes. They consist of glycerol molecule esterified into two fatty acids and a phosphate group. The phosphate group can be further modified with other groups such as choline or serine. Phospholipids have a polar head and non-polar tail, which allows them to form a lipid bilayer in cell membranes. Sphingolipids are a class of lipids that contain sphingos and backbone instead of glycerol. They are found in cell membranes that are involved in cell signaling and recognition. Sphingolipids can be further divided into several subcategories, including sphingomyelins, ceramides, and gangliosides. Steroids. Steroids are a class of lipids that are characterized by a four-ring structure. They are important in maintaining the structure and function of cell membranes, and they also act as hormones and signaling molecules, and they are four-ring structure. Examples of steroids include cholesterol, testosterone, and estrogen. These are hormones. And next, waxes. Waxes are a type of lipid that are esters of long-chain fatty acids and long-chain alcohols. They are used by plants and animals for protection against water loss and other environmental stresses. In summary, lipids can be classified into several categories based on their classic chemical structure and properties, including fatty acids, triglycerides, phospholipids, sphingolipids, steroids, and waxes. Each of this type of lipids, we will be once again quickly reading about the waxes. Waxes are a type of lipid that are esters of long-chain fatty acids and long-chain alcohols. They are used by plants and animals for protection against water loss and other environmental stresses. And that's it. So each of these types of lipids have different functions in living organisms that are important for maintaining cellular structure and function. Simple lipids, fats, oils, waxes, compound lipids, and glycolipids. Now we have to read everything about this in detail. Simple lipids. Simple lipids, also known as neutral lipids, are esters of fatty acids and alcohols. They can be further classified into three subcategories like fats. Fats are a simple lipid that are solid in room temperature. They are typically found in animals and are used in energy storage, insulation, and cushioning of internal organs. Oils. Oils are simple lipids that are liquid at room temperature. They are typically found in plants and are used for energy storage as well as for lubrication and protection against water loss. Then we have waxes. Waxes are simple lipids that are esters of long-chain fatty acids and long-chain alcohols. They are typically found in plants and animals and used for protection against water loss and other environmental stresses, like in leaf structures, in apple surface, everywhere. Then compound lipids. Compound lipids are lipids that contain other types of molecules in addition to fatty acids and alcohols. They can be further classified into several subcategories like phospholipids. Phospholipids are compound lipids that are important components of cell membranes. They consist of glycerol molecule esterified into two fatty acids and a phosphate group. The phosphate group can be further modified with further another groups such as choline or serine. Phospholipids have a polar head and non-polar tail, which allows them to form a lipid bilayer in cell membranes. Next, glycolipids. Glycolipids are compound lipids that contain carbohydrate group in addition to fatty acids and alcohols. Wait a minute, what was Phospholipids are compound lipids that are important components of cell membranes. They consist of a glycerol molecule esterified into two fatty acids and a phosphate group. The phosphate group can be further modified with other groups such as choline or serine. Phospholipids have a polar head and nonpolar tail, which allows them to form a lipid bilayer in cell membranes. Next, glycolipids. There will be definitely a carbohydrate portion, but that is not present in phospholipid, right? Yes, that part is not present in phospholipid. That is only present in glycolipid. They are important components of cell membrane and are involved in cell recognition and signaling. Next, lipoprotein. Lipoproteins are compound lipids that contain both lipids and proteins. They are important for the transport of lipids in the bloodstream and are involved in the regulation of cholesterol levels in the body. Next, sulfolipids. Sulfolipids are compound lipids that contain a sulfate group in addition to fatty acids and alcohols. They are found in bacteria and are important for their survival in harsh environments. Then there will be derived lipids. Derived lipids are lipids that are formed from the modification of other types of lipids. They can be further reclassified into several subcategories like steroids. Steroids are derived lipids that are characterized by a four-ring structure. They are important in maintaining the structure and function of cell membranes, and they also act as hormones and signaling molecules. Examples of steroids include cholesterol, testosterone, and estrogen. Then there will be eicosanoids. Like eicosanoids are derived lipids that are involved in inflammation and other physiological process. They are formed from arachidonic acid, a fatty acid found in cell membranes. Retinoids are derived lipids that are important for vision and skin health. They are formed from vitamin E, a fat-soluble vitamin found in animal products. In summary, lipids are a diverse group of biomolecules that can be classified into several categories based on their chemical structure and properties. Simple lipids include fats, oils, and waxes, while compound lipids include phospholipids, glycolipids, lipoproteins, and sulfolipids. Derived lipids include steroids, eicosanoids, and retinoids, among others. Each of these types of lipids have different functions in living organisms and are essential for maintaining cellular function and integration. So, biological roles of lipids as food reserves. Next question is about biological roles of lipids as food reserves, structural lipids membranes, as cofactors, as pigments, as insulators, as vitamin carriers in detail. So this is about biological roles of lipids. Earlier we have learnt about the classification like simple, compound, derived lipids. In case of simple lipids, we have learned that fats, oils, and waxes are there. In case of compound lipids, phospholipids, glycolipids, lipoproteins, and sulfolipids are there. And in case of derived lipids, we have learned about steroids, eicosanoids, and then retinoids. And now we are moving on to the functions or the biological roles. Lipids have many biological roles in living organisms. Here are some of the main roles. Food reserves. Simple lipids such as fats and oils serve as energy stores in living organisms. They can be broken down into their constituent fatty acids and used as a source of fuel when glucose is not available. In animals, fats are stored in adipose tissue. So when we look at the point of food reserve, simple lipids such as fats and oils serve as energy stores in living organisms. They can be broken down into their constituent fatty acids and used as a source of fuel when glucose is not available. In animals, fats are stored in adipose tissue, while in plants, oils are stored in seeds and fruits. Structural lipids in membranes. Phospholipids, which are a type of compound lipid, are a major component of cell membranes. They form a lipid bilayer that separates the interior of the cell from the extracellular environment. The hydrophobic tails of the phospholipids face inward, while the hydrophilic heads face outward, creating a barrier that is selectively permeable to certain molecules. Other lipids such as cholesterol and sphingolipids also play a role in maintaining membrane structure. Cholesterol and sphingolipids also play a role in maintaining membrane structure and function. Then cofactors. Some lipids such as phosphatidylcholine and phosphatidylserine act as cofactors in enzymatic reactions. They can bind to enzymes and modulate their activity, allowing them to function more effectively. Which are they? Phosphatidylcholine and phosphatidylserine. Then pigments are there, like some lipids such as Carotenoids serve as pigments in plants and animals. They absorb light in the blue and green regions of the visible spectrum, reflecting light in the yellow, orange, and red regions. This gives plants and animals their characteristic color and can also protect them from damage from UV radiation. Insulators. Some lipids, such as myelin, serve as electrical insulators in nerve cells. We know that myelin sheath is there in nerves. So myelin is a specialized lipid-rich membrane that surrounds nerve fibers and allows for rapid transmission of electrical impulses. Vitamin carriers. Some vitamins, such as vitamin E, vitamin K, serve as carriers for fat-soluble vitamins. These vitamins cannot be absorbed by the body without lipids, and they play an important role in many physiological processes. So when we look at the food results, there are many storage lipids, like something which is related with seeds. We will be storing it in seeds, like fats and oil. Then structural lipids, phospholipids, and some other lipids like cholesterol is also a structural lipid, and sphingolipids also play a major structural role. Then cofactors such as phosphatidylcholine and phosphatidylserine act as cofactors in enzymatic reactions. Then pigments. Carotenoids serve as pigments by absorbing visible light and in blue and green regions of visible spectrum, reflecting light in the yellow, orange, and red regions. Next, insulators. Myelin sheath. Just remember myelin sheath and just remember the electrical impulses along with that. Then vitamin carriers. Some vitamins such as vitamin E and K are other vitamin carriers. So that's about biological roles of lipids. In summary, lipids have many important biological roles in living organisms, including serving as energy stores, forming the structural component of cell membranes, acting as cofactors in enzymatic reactions, serving as pigments, acting as electrical insulators, and carrying fat-soluble vitamins. These functions are essential for maintaining the structure and function of cells and tissues and carrying out certain physiological processes. Next, we will be learning about the prostaglandins, chemical structure, nature, and functions in detail. So, before this, we have completed the biological roles in lipids as food reserves, storage lipids, structural lipids in membrane, as signals, as cofactors, as pigments, and insulators as vitamin carriers. We forgot the point about signaling. So, you will be learning about that later. Just don't forget the topic, lipid in signaling. So prostaglandins, chemical nature and functions in detail. Prostaglandins are a group of hormone-like substances that are derived from fatty acids, particularly arachidonic acid, which is found in cell membranes. They are characterized by a 20 carbon chain and a cyclopentane ring, which gives them a unique shape. Arachidonic acid is there and cyclopentane shape is there. And prostaglandins are synthesized by a variety of cells in the body, including platelets, endothelial cells, and inflammatory cells. They act locally and are rapidly metabolized, which allows them to have a short half-life and a specific effect. Prostaglandins have a wide range of functions in the body, including inflammation. Prostaglandins are involved in the body's response to injury and infection. They cause inflammation by dilating blood vessels and increasing blood flow to the affected area. This allows immune cells to reach the site of injury and fight infection. Pain. Prostaglandins stabilize nerve endings to pain. Prostaglandins sensitize nerve endings to pain, which can cause pain and discomfort at the site of inflammation. Fever is associated with that. Prostaglandins are involved in the body's response to fever. They act on the hypothalamus, which regulates the body's temperature to raise the set point and cause a fever. Blood clotting. Prostaglandins play a role in blood clotting by stimulating the aggregation of platelets and promoting the formation of blood clots. Reproduction. Prostaglandins are involved in various effects in reproduction, including ovulation, fertilization, and pregnancy. They can cause the contraction of the uterus and facilitate the movement of sperm and egg cells. Then gastrointestinal function. Prostaglandins play a role in maintaining the integrity of the gastrointestinal lining and regulating the secretion of mucus and bicarbonate. They also stimulate the contraction of smooth muscles in the intestine. Respiratory function is also performed by prostaglandins. Prostaglandins are involved in regulating airway diameter and mucus production in the lungs. They can cause the constriction or dilation of airways depending on the type of prostaglandin. In summary, prostaglandins are a group of hormone-like substances that are derived from fatty acids and have a wide range of functions in the body. They play a role in inflammation, pain, fever, blood clotting, reproduction, gastrointestinal function, and respiratory function. We'll be just once again reading what is the gastrointestinal function. Gastrointestinal function, that is when we just look at the gastrointestinal function, integrity of the gastrointestinal lining, maintenance and regulating secretion of mucus and bicarbonate secretion is associated with that, and along that the contraction of smooth muscles in the intestine is also associated with it. And respiratory function, airway passage diameter, airway diameter, and the mucus production in the lungs. Same is the case with constrictions and dilation of airways also happening. So that's about prostaglandins. By understanding the chemical nature and functions of prostaglandins, scientists can develop drugs that target specific prostaglandin pathways to treat various diseases and conditions. Next is about fatty acids, definition and essential fatty acids. Fatty acids are organic molecules that consist of a long hydrocarbon chain with a carboxyl group at one end. They are a major component of lipids, including oils, fats, and phospholipids. Fatty acids can be classified based on the length of their carbon chain and degree of saturation, like the presence of double bond or single bond, and the position of the double bonds. Saturated fatty acids have no double bonds, while unsaturated fatty acids have one or more double bonds. Monosaturated fatty acids have one double bond, while polysaturated fatty acids have two or more double bonds. So essential fatty acids are there, and they are fatty acids that cannot be synthesized by the body and must be obtained from the diet. The two main types of essential fatty acids are omega-3 fatty acids. The two main types of essential fatty acids are omega-3 fatty acids and omega-6 fatty acids. These omega-3 and omega-6 fatty acids play important roles in the body, including maintaining the integrity of cell membrane, regulating inflammation, and promoting healthy growth and development. Omega-3 fatty acids such as EPA and DHA are found in oily fish such as salmon, mackerel, and sardines, as well as in flax seeds, chia seeds, and walnuts. They are important for brain function, heart health, and reducing inflammation. Omega-6 fatty acids such as linoleic acid and arachidonic acid are found in vegetable oils, nuts, and seeds. They are important for skin and hair health, regulating inflammation and supporting the immune system. In summary, fatty acids are organic molecules that are a major component of lipids. Essential fatty acids such as omega-3 and omega-6 fatty acids are fatty acids that cannot be synthesized by the body and must be obtained from the diet. These fatty acids play an important role in maintaining health and preventing diseases. Omega-3 and omega-6. Next topic is about classification with examples: saturated, unsaturated, hydroxyl, and cyclic fatty acids. Fatty acids can be classified based on various characteristics such as their degree of saturation, presence of functional groups, and carbon chain length. Here are some examples of different types of fatty acids. Saturated fatty acids. Saturated fatty acids have no double bonds in their carbon chain, which make them more stable and solid at room temperature. Examples include palmitic acid, found in palm oil and animal fats, and stearic acid, found in animal fats and cocoa butter. They are soluble, stable, and solid at room temperature. Palm oil and animal fat and stearic acid found in animal fats and cocoa butter. Next is the unsaturated fatty acids. Unsaturated fatty acids have one or more double bonds in their carbon chain, which make them more liquid at room temperature. Examples include oleic acid. As they are unsaturated, that's why they just see like liquid at room temperature. Examples include oleic acid monosaturated, found in olive oil and nuts, and linoleic acid. That's a polyunsaturated one found in vegetable oils and nuts. Next, hydroxyl fatty acids. Hydroxyl fatty acids have a hydroxyl OH group attached to their carbon chain, which makes them more polar and soluble in water. Examples include ricinoleic acid, found in the castor oil, and hydroxyoctadecanoic acid, found in some plants and insects. Cyclic fatty acids. Next classification is cyclic fatty acids. Cyclic fatty acids have a ring structure in their carbon chain, which make them more stable and resistant to oxidation. Examples include cyclopropane fatty acids, found in some bacteria, and cyclohexyl fatty acids, found in some plants. In summary, fatty acids can be classified based on Their degree of saturation, saturated versus unsaturated, presence of functional group hydroxyl fatty acids, and carbon chain structure like cyclic and fatty acid chains. Understanding the different types of fatty acids is important in understanding their properties and functions in biological systems. Next, nomenclature of fatty acids, Genevan system in detail. The Geneva nomenclature system is a widely used method of naming fatty acids. It is based on the number of carbon atoms in the chain, the number of position of double bonds. First, we will be dealing with the basic points about this Genevan system. So, Genevan nomenclature system of fatty acids, widely used method of naming fatty acids, is based on the number of carbon atoms in the chain, the number and position of double bonds, and the position of any functional group associated with the chain. Here is a step-by-step guide to using Geneva nomenclature system. Count the number of carbon atoms in the chain. It is indicated by a number such as 14 for a 14 carbon chain. Determine the number and position of double bonds. Double bonds are indicated by a superscript number followed by the location of the first double bond from the carboxyl end, for example 18, and indicating an 18 carbon chain with one double bond at the 9th carbon from the carboxyl end. Indicate any functional group on the chain. This is done by using a prefix of suffix, whatever it may be, depending on the type of the group. For example, hydroxy indicate hydroxyl group and it is written as OH at the appropriate position in the chain. Epoxy indicate the epoxide group, O group, and written as O at the appropriate position. Combine the information from steps 1 to 3 to create the complete name of the fatty acid. For example, 14O indicate a 14 carbon chain with no double bonds. That means that's saturated. Indicate an 18 carbon chain with 2 double bonds at the 9th and 12th carbon positions. Here are some examples. This sign indicates delta. Here are some examples of fatty acids named using the Genevan system. Palmitic acid, 16 carbon chain with no double bonds. Oleic acid, 18 carbon chain with one double bond at the 9th carbon position. One double bond at the 9th carbon position. Linoleic acid, 18 carbon chain with two double bonds at the 9th and 12th carbon position. Arachidonic acid at 20th carbon chain with 4 double bonds at 5th, 8th and 11th and 14th carbon positions. In summary, the Geneva nomenclature system is a useful way to name fatty acids based on their carbon chain length, double bond position and functional group position. Understanding the naming system is important for identifying and classifying different types of fatty acids in biological systems. So that's it about Genevan classification. Next, we are moving on to another major topic, that is the structural organization of DNA in a assay form. Like DNA, that is deoxyribonucleic acid, is a complex macromolecule that carries genetic information in living organisms. It is the fundamental basis of heredity and plays a vital role in the development and functioning of all living organisms. DNA has a unique structural organization that allows it to store, transmit, and replicate genetic information accurately. In this essay, we will be discussing about the major structural organization points related to DNA. DNA structure. DNA is composed of 4 types of nucleotides, which are the building blocks of DNA. Each nucleotide contains a sugar molecule, a phosphate group, and a nitrogenous base. The sugar molecule in DNA is deoxyribose, which is 5 carbon sugar. The 4 nitrogenous bases in DNA are adenine, cytosine, guanine, and thymine. This nitrogenous bases pair up with each other in a specific way. A always pairs with T, and C always pairs with G. This base pairing is essential for the accurate transmission of genetic information during cell division and DNA replication. This base pairing is essential for the accurate transmission of genetic information during cell division and DNA replication. The nucleotides are arranged in a linear chain with the phosphate group of one nucleotide linking the sugar nucleotide of the adjacent nucleotide. The phosphate sugar backbone of DNA molecule forms the outer edges of the double helix structure. The nitrogenous bases project inward, forming the trunks of the DNA ladder. The double helix. The linear chains of nucleotides and DNA twist around each other to form a double helix structure. The 2 strands of the helix are held together by hydrogen bonds between the nitrogenous bases. Adenine pairs with thymine through 2 hydrogen bonds, while cytosine pairs with guanine through 3 hydrogen bonds. Adenine to thymine 2 and guanine to cytosine 3. This base pairing requires and is very important, and it ensures that the two strands of the helix are complementary to each other. The double helix structure of a DNA provides several important advantages. First, it allows for the accurate transmission of genetic information during cell division and DNA replication. Second, it provides protection for the nitrogenous bases, which are sensitive to damage from the environmental factors. Wait a minute. This double helical structure of the DNA provides several important advantages. First, it allows for accurate transmission of genetic information during cell division and DNA replication. Second, it provides protection for the nitrogenous bases, which are sensitive to damage from environmental factors. Such as radiation and chemicals. Third, it allows for easy access to the genetic information stored in the DNA molecule by other cellular components. Next is the chromatin structure. In eukaryotic cell, DNA is organized into complex called chromatin. Chromatin is composed of DNA, histone proteins, and other proteins that help to organize and compact the DNA molecule. The histone proteins are positively charged and interact with the negatively charged DNA molecule to form a nucleosome. The histone proteins are positively charged and interact with the negatively charged DNA molecule to form a nucleosome. A nucleosome is composed of a core of 8 histone proteins around it. The DNA molecule is wrapped. The nucleosomes are often then organized into a more complex structure called a chromatin fiber, which can be further organized into higher order structures such as chromosomes. The organization of DNA into chromatin serves several important functions. First, it allows for the compaction of DNA molecule into a small space which is necessary for the DNA to fit inside the nucleus of a eukaryotic cell. Secondly, it helps to regulate the expression of genes by making certain regions of the DNA molecules more or less accessible to regular cellular components. It helps to protect the DNA molecule from damage by environmental factors. So in conclusion, DNA is a complex macromolecule with a unique structural organization that allows it to store, transmit, and replicate genetic information accurately. The double helix structure of DNA provides the basis for its stability and ability to transmit genetic information about cell division and DNA replication. The organization of DNA into chromatin is necessary for its compaction, regulation of genetic expression, and protection from damage. Understanding the structural organization of DNA is essential for understanding its role in the development and functioning of all living organisms.